Welcome to my second video of an overview of AbLead, an antibody lead analysis, selection, and developability engineering software platform. I'm going to log in to the platform as a demo user, and I'll jump right into an entry. You can see the dashboard characteristics and an overview of the rank ordering analysis in my first video. So as I covered before, this is a set of entries in a project with various attributes pre-computed and a scoring system to allow for rank ordering. But oftentimes it's beneficial to be able to do further analysis on a system like this. One of the first things that people like to do is an alignment, straight alignment. The system will align. I have a residue cutoff of 50, so it line wraps at 50 positions. It shows the entries that are in kappa, lambda, and then together as the heavy chains. And so it will group them automatically. You can click on an icon here to get both the linear and the mature linear sequence. Mature linear would be after a single peptide cleavage. And so this one starts at 1, as you can see. I can collapse that. The default for the system is IMGT numbering with north CDR boundaries. And so those are annotated on the set of alignments. There's a grid here that allows for rearrangement if you'd like to rearrange, and this would apply down in the heavy chains as well. So you can order the antibody entries as you see fit. If there are leader sequences, you could see those. You could see the constant domains if there are constant domains, or you can select all of those features and see all of them together. You can select different numberings or show all numbering simultaneously, and likewise all region depictions simultaneously. You can color the residues by acidic, basic, hydrophobic, aromatic, neutral, or deletion ABHand coloring or not. And I will show a consensus sequence that adds a consensus sequence to the alignment that is underneath that consensus. So separate for kappa, lambda, and heavy. And I'll fade the consensus so you can see where the residue differences are in this molecule. This is dynamic, so as I drag this, you can change the line length, or I can enter a value here, and it will snap to. You can look across this and see EU numbering in the constant domains. AHO includes this arginine. It's a split codon, or glycine, usually in lambda, as part of the framework. I agree, but IMGT does not. And so it shows that position is separate. There's kappa, there's the lambda, constant domains, and here are the heavy chain, CDR regions, numberings, and constant domains. If a residue is selected for evaluation, you can just select on a residue number and it draws a box. Simultaneously, you can do this with multiple entries as you wish. These are toggles, and so it helps you visualize where in a molecule your eye needs to focus for comparison. It's a pretty handy tool for an electronic lab notebook or for a report generation. You can export a numbering sheet. You can also export an alignment in Excel. When you export an alignment, that alignment is going to look like however you have it set up here. If you show leader sequences, it shows the leader sequences. If you ask for it to show constant domains, it shows constant domains. However you have the numbering and regions shown, it will show those. The only difference is that it will show the linear and mature linear numbering regardless of whether or not you have it open. And there's an Excel alignment view. The numbering sheet, let me download that again. It takes a little bit longer to compute, so I'll pause for a second. This is very nice for filing IP, for evaluation of positions, because it breaks out the antibody set into leader and regions within the framework as separate cells. And then for all of the molecules that were in the alignment, you can click on any one of them and get a view of linear, mature linear, the numbering systems, and the regions that are in the system. And these will all be shown regardless of how they're selected in a numbering sheet. So you can scroll through and see the numbering across all positions. Very helpful. But more than an alignment, sometimes an evaluation of the nearest neighbor is needed. And I showed in the last video that this is just a high level one-on-one nearest neighbor, and these are all fairly diverse, which is good. But I will pull up a clading analysis. You can select the initial clading distance. I'll just leave it at 5. You can select which chains you want to clade and which regions you want to use for clading. I'll just keep it at defaults and click update. It generates a tree. You can see that tree. I can click anywhere on the tree or drag this to change the groupings. So wherever this line cuts, below that line, those parts, those entries on the tree will be grouped together for an alignment view. It's showing the alignment however you have it selected in the regions. So for example, if I select just heavy chain CDR3, it will show just heavy chain CDR3 and the tree will reflect that. From this tool, you can export as well the alignment as an Excel file, and it will show whatever is showing in the alignment, and the cladogram as an SVG file, and it'll export this as an image in SVG format, so a vector format. So you could include this in an ELN or a report. So that's an initial view of how to visualize multi-sequence alignment and clustering. And in the next videos, I'll show some liabilities and positional frequency analyses and ways to evaluate these individual residues on sequence. I look forward to seeing you then.